one more on the screen. This will be the last from 2020. And we are already organizing the program for the new season, which will be released soon in Netflix. So today we will have with us Ricardo Franco-Duarte, and it's my pleasure to present him to you. So Ricardo has a degree in biology from the University of Porto and a Master of Molecular Genetics for the University of Minho, from the University of Minho, sorry. He was a brilliant student, and I think he was the first 20 we ever awarded in this course for a thesis defense. He finished his PhD in 2014, also in the University of Minho, and he has been with us since, working in European and national research projects. So I have known him from more than 20 years now, which is a long time. And it has been a pleasure and a privilege to have him working in our research group. During this time, he published 25 papers, so quite a very good production, 12 as first or senior author, which has been more recently, he has published some papers as senior author. And also he published 4 book chapters. He also supervised several students, 13 master and 11 BSc students. His research interests are diverse and include yeast diversity, bioinformatics, applied bioinformatics, sorry, and omics analysis applied to microbial diversity and evolution. You might have heard of him because he recently described a new yeast species and from vines in the Azores Islands. And several news came out and he gave some interviews for a while. So it has been talked about because of that. But that's not about that he's going to speak today. So let's give him the word. Ricardo.
Thank you, Professor Celia, for this kind introduction. Good afternoon, everybody. I want to start this presentation by thanking the organizers of these CBMA seminars for the invitation and for the opportunity to present my work. As you can see, I entitled my presentation YeastOmics, from tiny and ubiquitous organisms to biotechnological cell factories. In this talk, I will present some of the projects in which I have worked in the past years, and I chose YeastOmics as the title for this talk because it reflects 2 of the main areas of my research, yeasts and omics. As you will see, the majority of the results I will present here will conjugate these 2 areas. And so the outline of my presentation will start with an introduction about yeasts and about omics, especially because we have a very diverse audience here with different backgrounds. And so I will review some key concepts that are important to understand the results I will present after. Following, I will focus very briefly in 4 experimental works, all of them using omics to characterize yeasts and to explore their biodiversity and their biotechnological applications. Starting then with the first introduction about yeasts. These microorganisms, as many of you know, belong to the kingdom fungi. They are eukaryotic and unicellular organisms that are at the moment more than 1,500 yeast species described as belonging to 150 genera. And we believe these numbers are already outdated. And nowadays we believe that this diversity associated with yeasts are even larger. Despite being great models for research, as you will see further, yeasts have also associated an increased biotechnological importance, since they are used in many biotechnology industries, as I will show you soon. The use of yeasts in industries have associated a multi-million euro potential. From the group of yeasts, Saccharomyces Cerevisiae is by far the most known species, and it is also the mankind's oldest domesticated organism. And for this, we consider it to be the world's premier commercial microorganism for biotechnological applications. The great importance of this yeast led it to be the first eukaryote genome to be sequenced, and this sequencing project was published in 1996 by Goffo. And here we have the original research article in which the sequence of the 6,000 genes of Saccharomyces cerevisiae were published. And several years later, we are already publishing papers in which not one, but 1,011 genomes are analyzed at the same time. And this is from 2018. In 2020, we believe this number was already largely surpassed. Together with the genome, also the transcriptome, the proteome, and the metabolome of this organism has been revealed in past years, which makes this yeast a great model to be used in research. In terms of biotechnological importance, there are several areas in which yeasts have been used with remarkable results, being the most important one, as you know for sure, the fermentation industries. As you know, Saccharomyces cerevisiae is the yeast used to ferment wine and bread. But yeasts have also a plenitude of other applications, such as, for example, waste utilization. They are used for in biomedical research for to study drugs metabolism and to perform genotoxicity screens, in healthcare to test pharmaceuticals, to name just a few. Moving now to the omics part. As you know, the term omics refers to the comprehensive analysis of particular biological systems, and we use the suffix om or omics to refer to a set of things that are associated to a particular system's biology. There are hundreds of different omics terms, but the most important and the most used ones are here described in this figure: genomics, transcriptomics, proteomics, and metabolomics. And the arising of all these omics started with the publication of the human genome sequence. Here are the Science and Nature papers reporting the publication of the human genome. And since then, a large quantity of data started to be produced, which led to the rising of new methods to study and to analyze systems biology. Nowadays, a large complexity of omics terms have been suggested, and complex workflows are being published to obtain an holistic view of what is happening in each organism at a certain moment. In this way, and to show you how in our group we applied this omics knowledge, I move now to the first set of results in which we attempted to characterize, using omics approaches, a yeast collection. The complexity of omics terms was already very high, but since we like complexity, we suggested a new term, a new omics called phenometabolomics. This new term, suggested by our group, as you can see by the origin of the word, consists in the study of the relationships between the genotype and the phenotype of a particular organism, a field known as phenomics, and then the establishment of relations with the metabolic data. To study phenometabolomics, and this is also common for other omics, we need two important requisites. We have to use high throughput methods with the purpose to obtain always a high quantity of data. And we need then bioinformatic and mathematical approaches to analyze and combine this data. In order to perform the phenometabolomics characterization, we first constituted our strain collection, composed by 172 Saccharomyces cerevisiae strains, obtained from 25 countries worldwide that are here marked in this map, and belonging also to a spectra of 10 technological applications, 10 technological groups. We have in our collection strains used for wine fermentation, strains related with beer, with bread, clinical strains, strains isolated from the environment, etc. With this set of yeasts, we developed several experimental works throughout many years. In a first phase, we characterized these yeasts using a set of 30 phenotypic tests, all of them with biotechnological relevance. We related this phenotypic diversity with differences in the genome of the strains. We then performed individual fermentations and analyzed the metabolic profile of the strains. We connected these differences with differences in the transcriptome of the strains. And we also used other less invasive and modern techniques to obtain a complete vision of what was happening during the fermentation performed by these isolates. All these works took place during many years, as I told you, and several publications were released by our group for each of these characterizations. I will not enter into much detail about these results, mainly because I already present them here in CBMA before, but I chose one key result to highlight this part of the work. Here we have a schematic representation of all the data that we obtained after the completion of the phenometabolomic characterization of our strain collection. And at this point, we had data from several experiments, from several origins with multiple scales, and considering a high number of strains. And we need at some point to develop new methods to allow the fusion of these data sets in order to be able to study their correlations. Then, during many months, we developed several computational methods, and with them, we were able to study something called multidimensional methods, multidimensional models. This consists in small sets of data that combines information from several origins and that show different levels of correlation between them. I have here one of those models as an example, and in this example we can see that this model, this particular model that I'm showing as an example, tells us that we have information for 4 strains. These 4 strains have diverse technological origins, but they share a common phenotypic profile. They share common information, in particular this phenotypic profile, a particular genotype, and also concentration of some metabolites that are produced during fermentation and that were quantified by HPLC and by GCMS. But what is then the main potential of these models, of these multidimensional models? To give you just an example, imagine that you are a winemaker and that you are interested in produce a particular wine with a particular characteristic. As you know, yeasts have a great importance in winemaking because they produce a lot of compounds that will change the aroma and the flavor of the wine produced. So for this particular example, after the request of the winemaker, you can go to one of these models, for example, this one that I'm showing here, and you know that this model has associated a high concentration of ethyl butanoate. This is an ester that have associated an aroma of pineapple, and you can see that these metabolites is associated with a particular genotype. So since nowadays yeast collections have all their isolates characterized genetically, you can go to these collections and choose the strains that have this particular genotype. And you know that they have the great potential to produce a wine with a pineapple aroma. You can imagine that this could save a lot of money to the winemakers, and it can also fasten a lot the choice of the best yeasts for winemaking. Moving now to the second work that I had included in this talk, it respects to the genomic variation during adaptation. This work started having as basis another work from our group that showed that isolates of a particular commercial wine yeast, the Zimaflor VL1, that are used commonly in a particular winery in the Portuguese demarcated wine region, earmarked with a W, could be isolated from the nature in locations that go up to 200 meters away from the original winery when this strain was being used. All of these isolates, here marked with different colors, were identified as being isogenic isolates of the same strain as the one used in this winery, the VL1 strain, and that were spread in nature around the winery during several years. And in the present work, we plan to understand to what extent does the genome of these isolates evolved in order to allow these isolates to adapt to the conditions found in nature. So for this, we chose 4 isolates that we found previously to be heterogeneous for some genetic markers, and performed several analyses with them, as I will show you next, always in comparison with the mother strain, the VL1 commercial yeast that was used as reference. In this way, our objectives for this work were to characterize the genome of these isolates and connect the detected changes to alterations in their phenotypic and metabolic heterogeneity. The ultimate goal of this analysis would be to conclude about the adaptive mechanisms that are happening during the isolate's permanence in the vineyard environment. The first technique we used to analyze the genome of these isolates was a technique of DNA microarrays that detect changes in gene copy number. And here in this image, we have represented the first 8 chromosomes of Saccharomyces Cerevisiae, plus the mitochondrial one, and also the locations of the TY elements in green. These TY elements are retrotransposons that have a mobile location in the genome and are many times associated with changes in the expression, in the gene expression in these yeasts. So results shown here by these orange symbols show significant changes in terms of gene copy number between the 4 analyzed isolates, always in comparison with the reference mother strain. And we can see that these changes have a stochastic distribution throughout various chromosomes. They correspond to 6 amplified genes in these first 8 chromosomes and to 6 amplifications coincident with TY elements. Some of these amplification events are common to results obtained by other authors that also detect them as being associated to wine strains. A similar scenario was found for the remaining 8 chromosomes, and in summary, we obtained with this analysis a total of 14 genes with an amplified number of gene copy numbers, mainly related with mitosis, with meiosis, with lysine biosynthesis, with galactose catabolism, and with asparagine catabolism. The amplification of this gene, ASP32, has an important role since it is described as being expressed in conditions of nitrogen starvation. And this is very important because it is related with the conditions that these strains are facing during their adaptation to the natural conditions, in particular to conditions with lack of available nitrogen. Besides changes in gene copy numbers, we found also a high number of differences in the nucleotide sequence of the DNA of these isolates. I will not enter in much detail because I don't have time, but we were able to detect several hundreds of differences already imprinted in the genome of these isolates that were not detected when we analyzed the mother strain, the reference mother strain. Besides differences in the genome, we were able to detect also a distinct phenotypic pattern between these isolates, with differences in 14 out of 30 phenotypic tests. We found also metabolic differences when analyzing by HPLC the production of 3 compounds, malic acid, glycerol, and ethanol. And finally, we detected also differences in the GCMS analysis, in particular in production of 3 aromatic compounds, benzene ethanol, 2-methyl-1-butanol, and isobutanol. All these differences were detected in comparison with the reference mother strain, don't forget, which indicates that these natural isolates have already a different metabolic pattern. And even if used to produce wine, they would for sure contribute with a different aromatic pattern. So in summary, the main outputs of this work were that the isogenic isolates of the commercial strain Zimaflor vel1 have already genomic differences after their presence in nature during many years. These genomic differences could be already extended to the phenotypic and to the metabolic part, as detected by the variability in their profiles and in the metabolites quantified by HPLC and GCMS. And the main take-home message here is that the transition from nutrient-rich musts to nutritionally scarce natural environments is correlated with microevolutionary changes that are happening during the permanence of these isolates in nature, and that reflect their adaptive response to those conditions. The following set of results I want to show you relates with the use of yeasts as microbial cell factories. In this particular case, I will show you results of a publication of our group in which we characterize the capacity of a set of yeasts to produce succinic acid. Starting with a brief introduction about succinic acid, this acid is a platform chemical with an important role as precursor for the synthesis of biodegradable polyesters, resins, pharmaceutical, and even as food additive in many industries. Nowadays, succinic acid is still produced by a process that has as basis the petroleum, which implies, as you know, high costs for the environment. In this way, the biological production of succinic acid is gaining high attention lately, using microorganisms to produce it and using renewable resources. This biological production has been tested using two groups of organisms, bacteria and yeasts, both with reasonable results. However, the production of succinic acid, the bioproduction of succinic acid using yeasts have some advantages. For example, the manipulation of yeasts can be made using lower pH values in comparison with bacteria, which leads to an improvement in the overall process, and consequently it allows to lower the costs of this purification process in opposition to the use of bacteria that carries an increased cost in this purification parts. With this in mind, the main objectives of this work were to develop methods to identify promising natural Saccharomyces Cerevisiae yeast isolates that show already some capacity to produce succinic acid, to characterize these isolates using genomic and transcriptomic approaches, and with this knowledge to understand the key factors that contribute to reveal this potential ability of the natural isolates to produce succinic acid. And with this, we can in the future design new strategies to perform genetic manipulations, leading to a high production of succinic acid. To achieve these objectives, we started with a set of 17 well-characterized Saccharomyces Cerevisiae strains. We subject them to individual fermentations in conditions that were previously optimized to be adequate for succinic acid production. And by HPLC, high performance liquid chromatography, we quantified the production of succinic acid by these 17 isolates, obtaining highly diverse results, as you can see here in this image. But this succinic acid production varied between almost zero in this case and reached a maximum of 1.13 grams per liter. And although this value is still low, it is already considered that satisfactory, considering that we are using natural isolates, not engineered ones. And this production was already at the same level as the one obtained when using genetically manipulated strains. For further analysis, we chose the two best producers, here marked in green, a strain with no production, and an intermediate producer, to perform a comparative analysis between these two groups of strains. After the whole genome of these isolates was sequenced using next generation sequencing platforms, and the genomes were characterized in terms of SNPs, single nucleotide polymorphisms, that are differences in a single nucleotide when comparing two DNA sequences, in terms of indels, that are small insertions or deletions in the genome, and also in terms of gene copy number variations. In other words, changes in the copy number of a particular gene that vary between individuals, or in this case, between groups of strains. Analyzing first the presence and distribution of SNPs per chromosome, we can see here the number of SNPs quantified for the good producer strains, here marked in green, and for the non-producer strains, here marked in blue. Analyzing the 16 yeast chromosomes, the 16 Saccharomyces Cerevisiae chromosomes, we can see that there are already some point differences in some parts of some chromosomes. However, no clear pattern of differences can be detailed. To particularize this analysis and to find other types of differences, we selected 42 key genes that are here described in this table, that included also transcription factors. And these genes were selected because they have an important role in the succinic acid production pathways that are here represented in this figure. From this list of 42 genes, 31 showed already a change profile that are here represented in terms of the presence or absence of SNPs in the genome. We're comparing between two groups of strains. And these changes that were detected in these 31 genes correspond to changes that lead to changes in the DNA reading frame. And from these 31 genes, 23 marked in yellow represent genes that lead also to an amino acid of a different functional category, which increased, of course, their potential role as explaining differences between both groups of strains, the good producers and the non-producers. In order to understand if these changes were also extensible to higher or lower number of copies, of gene copies, we analyzed these hypotheses in the whole genome of these isolates. And we have here represented these results, again, for the 16 yeast chromosomes, considering in green the neutral case, in which no copy number changes were detected, and in blue and red, cases in which there were a decrease or increase in the copy number profile. Results show that strains with low capacity to produce succinic acid, here on the left, showed an increased number of copies of genes present in chromosomes 1 and 11, in opposition to strains with higher capacity to produce succinic acid, here on the right, that showed no alteration in these chromosomes or in any other chromosomes. After this genomic characterization, we select a set of 10 genes that showed higher fluctuations in the previous analysis and that are considered as important in the succinic acid production pathway. Then we performed a transcriptomic analysis in 2 fermentation time points, comparing always the good and the poor producers of succinic acid. These results, in comparison with the ones obtained in the comparative genomic analysis, allowed us to understand the mechanisms that underlie the differences between these 2 groups of strains. In the future, these results could give us clues to draw strategies in order to improve and engineer yeast strains with the purpose to produce higher amounts of succinic acid. Very briefly, because time is flying, we have here a schematic representation of the results obtained in this transcriptomic characterization with the 10 chosen genes. On the left, we have the non-producer strains, and on the right, the good producer strains. In red, I represented the genes in which a higher expression was detected, and in green, the genes that were detected as sub-expressed in the end of fermentation. The main results of this analysis showed for the non-producer strain a higher expression of the genes SDH1 and SDH2, which explains a degradation of succinate to fumarate, and in this way explains why there is no accumulation of succinates in these isolates. And also a higher expression of ABH genes that are involved in the ethanol production, and that explains also a redirection of the metabolism of these isolates towards the production of ethanol instead of being redirected to the TCA cycle, which would lead to an accumulation of the succinate production. One new result obtained in this work was that was not yet reported before, was the detection of a higher expression of the transcription factor AP4. This transcription factor is known as being involved in the dioxic shift. There is to say, in a shift between the formation of byproducts towards the biomass formation and growth. And we speculate that the expression of this transcription factor could be an important factor to be analyzed in higher detail in the future, since if we can eliminate its expression, we can redirect yeast metabolism to metabolites production instead of being directed to growth and to production of biomass. In conclusion, the main outputs of this work were that we could already obtain a relevant amount of succinic acid as being produced by natural isolates. We already detected some candidate SNPs that can be in the basis of the differences found between the good and the bad producers regarding gene differences. In transcriptomic analysis, by transcriptomic analysis, we detected also new candidate genes and transcription factors to be analyzed, to be detailed, analyzed in the future. And in this way, and to wrap up, all this information allowed us to obtain an holistic view of the key factors that contribute to reveal the potential ability of natural isolates to be used as successful producers of succinic acid, and this knowledge could lead in the future to the design of new engineered strains. The last set of results I have planned for you today relates with the evolution of yeast mitogenomes. This is a recent work. It was published this year. And in this work, we attempt to study the Saccharomyces Cerevisiae mitogenomes. In other words, we want to use mitochondrial DNA to analyze the evolution of Saccharomyces Cerevisiae and to understand what kind of information we could get from the mitogenome that we couldn't obtain from the nuclear genome. But why use mitochondrial DNA for this? The main reason for this is because at least in humans and in other higher eukaryotes, we know that the mitochondrial DNA appears to not recombine. This is to say that you inherit your mitochondrial DNA from your mother, and it goes without many alterations for the following generations. So this is great to analyze evolution and to understand how a particular species evolved a long time, but does this happen also in yeasts? That is what we wanted to understand in this work. Regarding Saccharomyces Cerevisiae evolution and domestication, there are several studies, all using nuclear genome, nuclear information, that place the origin of Saccharomyces Cerevisiae in China. And then populations were merged with wild lineages from other regions in Asia and in other parts of the world. And these authors suggested that this evolution must have been based in adaptation to high sugar environments. So this is a strong evidence already to the fact that these domestication events didn't follow a particular route in the world, but instead these events were related with the type of environment that these yeasts were facing, and if this environment in particular was rich in sugar or not. So the question that became always present when we started to study yeast evolution, and in particular how the domestication of Saccharomyces Cerevisiae took place, was why is it so hard to analyze yeast evolution and domestication? In particular, why there is so large discrepancy between the so large number of studies explaining human evolution using mitochondrial DNA that have been published during so many years, as you are seeing, and the ones using Saccharomyces Cerevisiae mitogenomes that are almost inexistent. We came to the explanation for this during our work. The main cause for this is that in Saccharomyces Cerevisiae, in opposition to what happens in humans and in higher eukaryotes, the mitochondrial DNA recombines. The mitochondrial proteins and parental genomes mix in the same partition. There are biparental transmission leading to recombination, And this recombination happens mainly in the intergenic regions. So this was for us at the beginning, as you can imagine, a cause of great frustration, and we almost quit this work. But luckily, and jumping now a few months in time, we were able to build one approach to compare different yeast mitogenomes. And for this, we collected more than 12,000 mitogenomes from NCBY. And after many computational analyses, we built a pipeline in which we use only some parts of the mitochondrial DNA of Saccharomyces cerevisiae, here marked in red in this image. And by using only these parts of the mitochondrial DNA, we were able, for the first time, to compare between different strains. We were able to align the DNA of the different strains and perform new studies analyzing yeast mitogenomes. After establishing this pipeline, we applied it to a final data set of 6,646 Saccharomyces cerevisiae mitogenomes, for which we have information for their geographical origins and for their technological applications, and performed several population analyses to compare between different strains. Next, I will show you one example of the results we obtained with this population analysis. We performed several analyses using different computational methods, different bioinformatic methods, but I will show you only one example of the results we obtained. So using all the mitogenomes, we built phylogenetic trees, cataloging strains according to their technological origin, here in the top, and with their geographical origin, here in the bottom. Results show that some clusters were obtained containing strains from a single technological origin. However, no clustering was visible when considering strains' geographical country. Even considering FST values, here on the right, that is a measure of genetic divergence between populations, we can see a clear differentiation of laboratory strains, also as for the bred and clinical strains, clinical yeasts, which can already suggest that the history of Saccharomyces cerevisiae domestication must have had its basis in the adaptation to these particular niches. A particular aspect that we found is that wine strains presents also a moderate level of differentiation, suggesting an independent line of domestication, something that was also shown before using nuclear genomes. As main outputs of this work, we highlight that this work allowed us for the first time to build a pipeline to analyze Saccharomyces cerevisiae mitogenomes and to compare between different strains, surpassing the difficulties found correlated with the high recombination rate found in Cerevisiae mitochondrial DNA. With this work, we obtained clues about domestication of Saccharomyces cerevisiae that needs to be further explored. A great differentiation of laboratory, bread making, and clinical strains mitogenomes was detected. And finally, we found also differentiation of wine strains mitogenomes, suggesting that they follow a distinct event of domestication, which is in accordance with the same fact found using nuclear genomes. I finished this presentation by acknowledging all the people that contributed throughout many years to all the works that I presented here today, and also to the financial entities. And I thank you all for the attention given and for being here today listening to this talk. Thank you.
Thank you, Ricardo, for your presentation and for keeping to the time so tightly. So to give us time to have questions that probably arose from your presentation, where, focusing so many different subjects regarding omics of Saccharomyces cerevisiae. So I open the discussion. Questions, please. Master students.
It was all very clear.
There are no questions for Ricardo? I have. There is one question. Please, Carlos, turn off your microphone and your camera, please. So, I have.
Olá, conseguem-me ouvir?
Sim.
Sorry, I just wanted to know if in the genomic analysis you were able to identify SNPs that were important to improve the gene expression, and if some transcription factors were analyzed during this experiment, because I find it really interesting. Thank you.
Hello, Carlos. Clarify just one thing. You are referring to a particular work or just a global?
Yes. In the third work, you in the
about succinic acid.
Yes.
Succinic acid. Okay. Thank you, Carlos. Yes, we identified, as I told you, we performed several experimental analyses regarding the analysis of the genomes, and we detected several SNPs in genes related with the succinic acid production pathways. And we divided these analyses of SNPs, presence or absence, in several parts, because when you detect some SNP, you have to be careful about what that means. That's why I divided first the analysis of the 42 genes. And for that 42, I detected SNPs in 31 of them. And this is all already a relevant result, because for that 31 genes, these SNPs were responsible for changes in the reading frame of the DNA. But then for these 31 genes, 24 of them showed already changes that will lead to an amino acid of a different category. So for all the differences we found, these ones were the ones we considered as most relevant. So yes, we detected that SNPs you referred, and some of them were so important that it will lead to an amino acid of a different category. I hope I clarified your question.
If I may continue then, in those amino acid changes, did some express the significant role? Have you really tested that? Because I think that it can change the amino acid, but not the function of the protein, or improve the function. And I don't know if I'm correct.
Yes, that's actually a very good question. Thank you. Yes, we in terms of SNPs, we first did a global analysis, and then we particularized this analysis for these key genes that we know are involved in the succinic acid pathway. So all of these changes that I'm referring in these genes that lead to an amino acid of other category are all genes related and very specific for the succinic acid production pathway. And especially, as I showed after in the gene expression, those SDH genes and ADH genes, despite the fact that we detected changes in their expression in the transcriptomic analysis, we detected also before changes in terms of SNPs in these specific genes. So you are right, they are very important, and we hypothesize that these SNPs, together with the changes in the expression, are what are separating the two groups of yeast, the good producers and the bad producers. That's a very good question.
Okay, thank you. I think that's all.
Okay, thank you.
Thank you, Carlos. there is someone else. Yes, please turn on your camera and your micro.
Sure. Hello, can you hear me?
Yes.
Yes.
I have a question. I'm not sure if it's a good question or not, but in the presentation, you mentioned that you used the mitochondrial RNA and DNA for the yeast evolution, but I can't understand what is the point of using this instead of rRNA.
Okay, thank you for your question. I will try to clarify it. The point of using mitochondrial DNA, for example, it is very used in humans to study human evolution. As I show you by many examples there are hundreds and hundreds of papers published. Funny by one of the authors that published so many papers regarding human genome is one colleague of mine and of our center, Pedro Soares. And the reason of using mitochondrial DNA to study this evolution is because, at least in humans, since mitochondrial DNA does not recombine, you can detect by analyzing, for example, your mitochondrial DNA, you can go back to several generations. You can go several generations back and try to understand the pathway of evolution of the human race, for example. Okay, we try to do this with yeasts, but face the difficulties of found out that mitochondrial DNA in yeasts, and here particularizing Saccharomyces Cerevisiae, because we still don't know yet if other yeast species have the same procedure, but especially for Saccharomyces Cerevisiae, we found out that it recombines. So the first answer would be that for Saccharomyces Cerevisiae, we cannot use it the same way as we use it for humans. But that's why we then built this pipeline to be able to analyze it and to be able to compare different strains and do this procedure of going back to many generations to try to understand what were the roots of evolution and of domestication of Saccharomyces Cerevisiae. By the way, we want to do this in comparison of humans, if it's possible. So we are trying to do this, to use the same procedures as used in human evolution and in human genetics to yeast genetics in this case.
Okay, thank you. Thank you so much.
No problem.
Okay, any more questions? So, I'd like to ask you, not exactly a question, but what are your plans now?
Yes, so we have several works. Sorry?
What's next?
Yeah, we have several works now in place in laboratory. For example, that phenometabolomic characterization of Saccharomyces Cerevisiae strains. We are now trying to replicate it using another yeast species, Pterolachia delbrueckii, and we have several master students working in this work. And now our plan for the near future, our most ambitious plan is to continue the work related with succinic acid production. We are planning a project to continue that work, in particular to use Saccharomyces Cerevisiae always natural isolates, as in the work I showed, but this time we want to explore a technique called adaptive laboratory evolution. In detail, we want to evolve these yeasts in laboratory, subjecting them to several conditions that we know are adequate for the production of succinic acid. And in this way, we will force the arising of new mutations that can be advantageous to increase the production of succinic acid. In each round of evolution, of adaptive evolution, we want to sequence the genome of all the strains in each round, and then analyze those mutations that are arising at each round. And so, as you can imagine, this will be a great it will require a great effort of sequencing and a great effort of genome analysis to analyze and compare the mutations arising in each round of adaptive evolution.
Thank you. Lots of work ahead. If any more questions? So if there are no more questions, I thank you again, Ricardo,
thank you very much
for your presentation, and thank everybody for participating in this seminar, and hope to see you all next year, in the beginning of January, for our next Science on the Screen. Thank you.
Thank you.